Good afternoon. Today's lecture is the first in a series of 3 lectures dealing specifically with immune dysfunction. We've spent the entire semester building up our knowledge base and our understanding of how the immune system works when it is functioning properly. What we are going to transition to with the next 3 lectures is what happens when the immune system doesn't function the way that it's supposed to. So immune dysfunction, again, is just any type of condition where the immune system is not functioning properly. There are 3 broad categories of dysfunction that we will focus on. Today's lecture will deal specifically with hypersensitivity, which is an exaggerated immune response that tends to have deleterious effects and causes damage to the individual. We'll focus on this again for the topic of today's lecture. However, there are other categories of immune dysfunction. Autoimmunity is another category, and this is in fact an inappropriate response of the immune system to self-components. With both hypersensitivity and autoimmunity, there is a recognition issue. If you recall the first day of class, one of the first things I put on the board was that the immune system functions by recognition and response. And what happens in these 2 categories of dysfunction is that there is, in fact, a recognition problem. With hypersensitivity, the recognition is something typically environmental that is not truly a potential threat or a pathogen, and the immune system erroneously recognizes this external component. Autoimmunity, also a recognition problem. However, in this case, what is being recognized as a potential threat are the host's own molecules, proteins, components, and they are being recognized by the immune system, and an immune response is in fact initiated. With autoimmunity, we tie that together with a mechanism known as tolerance. We've alluded to tolerance a few times throughout the semester. Tolerance is a mechanism that exists to protect an individual from potentially self-reactive lymphocytes. So the tolerance mechanism is a safety mechanism to make sure that autoimmune situations don't in fact take place. So when tolerance fails is when autoimmune disorders come into play. So today's lecture focuses specifically on hypersensitivity. So let's break down that term hypersensitivity just to understand a little bit more about what's happening with this particular dysfunctional component of the immune system. It is in fact an inappropriate response. The term sensitive is defined as quick to detect or respond to slight changes, signals, or influences. And the root hyper means over, above, or excessive. So with hypersensitivity, we have a response that is above, over, excessive to very subtle changes or slight changes that can occur. So let's add some additional terms to this particular topic. The term prophylaxis has the word or root in it, phylaxis, which means protection against infection. So prophylaxis are measures that are designed to preserve health and prevent the spread of disease. These are protective or preventative measures to prevent disease. The term anaphylaxis is a term utilized in response to hypersensitivity. We'll discuss specifically what's happening with hypersensitivity. It is, in fact, a mast cell degranulation. However, the term anaphylaxis came into play because what was happening in individuals that were having a hypersensitivity reaction was that their reaction was against or opposite of prophylaxis. Again, measures designed to preserve health. These were things that were causing deleterious effects in the individuals in question. What was noted in early studies of hypersensitivity was that most individuals had, in fact, an inflammatory response that sort of went out of whack. With hypersensitivity, again, what is happening is something is being detected as a potential threat. That something is referred to as an allergen. And the allergen is usually something that is an environmental component, something not normally seen by the host system, but again, not truly a potential threat. However, it is being recognized as a potential threat, and what happens in the process is a degranulation event, which we will look at in more detail in just a minute. There are, in fact, 4 distinct types of hypersensitivity, and they are classified or categorized based on how the immune system responds to the allergen in question. Also, there are distinct effector molecules that play a role in each one of these classes or categories. Briefly, there is what is known as type 1, which is IgE-mediated. This is what we typically think of as a classic allergic response, and we will spend the most time discussing type 1 hypersensitivity. Type 2 hypersensitivity is antibody-mediated cytotoxic sensitivity, and in this particular case, antibodies are utilized in a situation to cause a cytotoxic effect, and we will look at this particular example in a little more detail as well. Type 3 hypersensitivity is known as immune complex-mediated, and we will spend a little bit of time discussing type 3. It turns out that type 1, type 2, and type 3 all involve humoral responses, which means antibodies are involved in all 3 types of hypersensitivity, type 1, type 2, and type 3. The last category or classification is type 4, which is known as delayed type of hypersensitivity, and this is a mechanism that depends on the activation of T cells and is not antibodies are not involved in this particular type of hypersensitivity. So let's start off talking about type 1 hypersensitivity, also known as IgE-mediated hypersensitivity. Obviously, in the title, the antibody class IgE is responsible for this particular response overall. So this slide is meant to be an overview, and we will look at more detail in the upcoming slides. What happens in this particular situation is that you do in fact have something known as an allergen. An allergen is in fact an antigen. It is usually something environmental, again, not truly a potential threat, but if you go back to the definition of an antigen is any type of molecule that can bind to an antibody and trigger or initiate an immune response. So an allergen is in fact an antigen in this particular case. What happens is the allergen binds to IgE antibodies that are specific for that particular allergen, and this eventually results in what's known as a degranulation of mast cells. So let's take a step back and review the IgE class of antibodies. Hopefully you recall that the IgE class of antibodies is a specific class of antibodies that plays a role in fighting off parasitic infections, in particularly against parasitic worms. So these IgE antibodies get produced with the ability to interact with the allergen that triggered the production of this particular class of antibody in the first place. These antibodies bind to Fc receptors on both basophils and mast cells. When the allergen comes in contact with the antibody that is bound to the Fc receptor on either basophils or mast cells, this triggers the degranulation event. So just to recap a little bit about what we know are found in the granules of basophils and mast cells, there are a number of different types of molecules, which again we'll look at in more detail in the next slide. In particular, histamine and heparin are key components that are being released in the degranulation event. Histamine, if you recall, acts in vasodilation as part of the inflammatory response, and heparin acts as an anticoagulant. Okay, so now let's take a little closer detailed look at what happens during type 1 IgE-mediated hypersensitivity. Up at the top of the slide, what you see is the typical exposure to the allergen, which then activates helper T cells, and those helper T cells stimulate B cells to form the IgE class of antibodies. So again, what you have is the allergen binding to the receptors on the B cell. There is then stimulation that occurs by the helper T cells to allow for the activation of these B cells to produce both memory cells and plasma cells. As we know, what plasma cells do is produce soluble antibody that is going to be secreted. And in this particular case, the antibody that is being secreted is the IgE class that now has the ability to bind to the original allergen that triggered this differentiation process in the first place. What happens next is the IgE antibodies bind to IgE-specific Fc receptors on the surface of mast cells and basophils. In this case, what you have now is a sensitized mast cell or a sensitized basophil cell. This is the very first exposure to the allergen. At this particular point, there is no release or degranulation that takes place. The degranulation event always occurs upon second exposure to that allergen. So after the first exposure, you get the sensitized mast cell or basophil cell. These are cells that are now sort of waiting for the next exposure of that allergen to then trigger the degranulation event. So next you would see upon a second exposure to this particular allergen, cross-linking of the allergen to the bound IgE, which then triggers the release of the vasoactive amines found within the granules. Here you can see a number of effects that occur because of those degranulation events. There are effects on smooth muscle cells, small blood vessels, mucus glands, blood platelets, sensory nerve endings, as well as eosinophils. So this slide is just showing us a little bit more of what happens after that second exposure to an allergen and the degranulation event. Across the center of the slide, you can see what happens as a result of the mast cell activation or degranulation. Cytokine production, chemokine production, growth factors, lipid mediators, and additional components all are stimulated in response to the degranulation event. The arrows from that box show you the cell types that are affected by all of these different components that have been released by the degranulation event. Most importantly, what we typically think of with an hypersensitivity reaction, specifically type 1, is inflammation as a response. So definitely hematopoietic cells, those blood cells that we know are part of the immune response, trigger to allow for excessive inflammation to take place. However, there are other changes that occur with different types of cells that occur again in response to the degranulation event. So allergens, again, are antigens that do in fact induce type 1 hypersensitivity. What you see in this slide are just common allergens associated with type 1 hypersensitivity. And if you look through the table, you can see some examples of things that you may be aware of as a typical allergen. Plant pollens, ryegrass, ragweed, for example. I know a lot of us locally suffer from allergies that occurred during the almond blossom. So again, there are lots of different examples of allergens that can in fact or are in fact associated with type 1 hypersensitivity. One of the things that I want to highlight at this particular point is that there are some examples where there's a clear-cut genetic component that allows for allergies to occur, meaning that the information is passed on from generation to generation that allows for these types of hypersensitivity reactions. However, that is a very small percentage of hypersensitivity reactions that can be defined or described by a genetic component, meaning that most individuals who suffer from allergies, we don't really have a clear understanding as to why this particular type of dysfunction occurs in these individuals. It turns out that there are, in fact, a number of ways that the degranulation event can take place. However, what I want to focus on specifically and for our purposes is the allergen cross-linkage that allows for the degranulation to take place. So what you see in this image is a typical mast cell with its Fc receptors. These Fc receptors are specific for the IgE class of antibodies. And in order for a degranulation event to take place, you have to have two antibodies bound to two receptors side by side, like you see in this image here, and the allergen interacting with both of the antibodies. This is referred to as cross-linkage, and it is this specific situation that allows for the triggering of the degranulation event. Again, there are other ways that degranulation can occur that you see on the slide, but for our purposes, we're going to focus on that panel A. Oftentimes, when discussing type 1 hypersensitivity or IgE-mediated hypersensitivity, we can discuss two broad categories of the type of effect found in an individual. The first one is known as a systemic anaphylaxis response. And in a systemic response, you have a shock-like, oftentimes fatal type of reaction that occurs within minutes of exposure to the allergen. These are typically things that enter directly into the blood, usually something that is absorbed by the gut or the skin. And again, why something reacts so dramatically in an individual is not necessarily clear, but there are in fact these clear-cut examples of what's known as systemic anaphylaxis. And there are extreme examples that we know of, individuals that have a peanut allergy. There are individuals who are allergic to bee stings and have to carry around what's known as an EpiPen, and an EpiPen contains a drug known as epinephrine. Epinephrine counters histamine by relaxing smooth muscle and reducing vascular permeability, and this oftentimes does in fact help the individual overcome this type of anaphylaxis. In a systemic response, typically what can happen is an individual can go into anaphylactic shock, where there's labored respiration, there's a drop in blood pressure, bronchial constriction, and eventual asphyxiation. So again, this is extremely severe, and individuals who have these type of allergies, again, often do carry around what's known as an EpiPen to be able to inject themselves if they do in fact come across this type of allergen, because oftentimes there's not enough time to get someone to the hospital when these types of reactions occur. Now, the more common type of type 1 hypersensitivity is known as local anaphylaxis, and these are the minor types of allergic reaction things that you may have heard about. Things like allergic rhinitis, where you have hay fever, sneezing. These are oftentimes nasal type of allergies. Individuals who have asthma is in fact due to a type 1 hypersensitivity. This does affect the respiratory tract overall. There are examples of food allergies like we touched on again with systemic anaphylaxis, but there are some that are much more mild with local anaphylaxis as well. And lastly, local anaphylaxis can also represent itself as atopic dermatitis, like a skin rash or a hive overall. Again, why individuals have certain categories of local anaphylaxis, that's not necessarily clear either. So what can be done to treat individuals with type 1 hypersensitivity? Well, the most common concept is to avoid contact with the allergen, which if you know you have an allergy to a specific component, to avoid the contact is in fact the best way to keep yourself healthy without having this type of response. However, sometimes that is not necessarily always possible, and so there are some drugs that are available that can help individuals with this type of hypersensitivity. Many individuals take antihistamines, and antihistamines block histamine receptors on target cells to then help with that excess response or excess release of histamine that occurs during the degranulation event. So we're not stopping the degranulation event, but hopefully we're blocking that histamine from binding to the target cells and playing the role that histamine would normally play. Epinephrine, like I mentioned just briefly, is going to stimulate something known as cyclic AMP, which then also plays a role in preventing degranulation. We know, again, it helps relax smooth muscle, and this can then reduce vascular permeability as well. Cortisone is another example of a drug that can be utilized to treat individuals with hypersensitivity. It is known to block the synthesis of histamine, and by blocking the synthesis of histamine, again, you can help alleviate some of the symptoms in these individuals. Okay, let's transition a little bit into type 2 hypersensitivity, which is known as antibody-mediated cytotoxic hypersensitivity. In this particular case, antibodies mediate destruction of cells due to activation of the complement system, acute inflammation, and initiating ADCC. So this, in fact, is a cellular type of response that occurs, meaning that antibodies can initiate the ADCC mechanism and play a role in destroying cells in that process. The last two classes of hypersensitivity are not nearly as common as type 1 and type 2, and so we're going to spend just a little bit of time talking about them briefly so that you have a general understanding of what's happening in these immune responses. Now, under normal immune function response, we know that antibodies can in fact form what are known as immune complexes. So in this slide, you can see the antibodies with the antigens as well as complement system components coming together to form these immune complexes. In normal circumstances, the job of these immune complexes is to bind up all of this antigen and eventually have it removed from the host system. In type 3 hypersensitivity, this is again a dysfunction. This is where this mechanism is not functioning properly. So these complexes form, and what happens is they're not cleared quickly enough from the host system. This then elicits extended periods of inflammatory response, which lead to tissue damage due to phagocytosis and lytic enzymes being released overall. So this is detrimental because these immune complexes are not cleared quickly enough, and the extended periods of inflammation are what cause the problem in these individuals overall. The last category of hypersensitivity I want to discuss briefly is type 4 hypersensitivity, which is known as cell-mediated hypersensitivity, also known as delayed type of hypersensitivity. It is a delayed type of hypersensitivity in that it usually takes 2 to 3 days to develop and also requires subsequent exposure with the component that is causing the hypersensitivity reaction in the first place. Now, this particular class, like I mentioned at the beginning of the lecture, is not antibody-mediated. There are no antibodies that are involved in this particular type of hypersensitivity. It is, in fact, a cell-mediated response. What happens is helper T cells recognize antigen bound to MHC class II. These helper T cells then release cytokines, which then cause an influx of cells to the target location with an increase in phagocytic activity. So what's happening is that you have a large influx of nonspecific inflammatory cells to the target location, and you are having an excess cytokine response as well as inflammatory response to this particular component. And lastly, with this particular slide, this is a table that gives you some examples of components that do in fact play a role in type 4 hypersensitivity. Things that you might be more familiar with are things like poison oak and poison ivy. These are in fact components that cause this category or class of hypersensitivity. The overall point with the 4 different classes is that they are individual reaction types, meaning they are distinct, separate types of reactions that are occurring in each one of the classes. The overarching theme for hypersensitivity, though, is something that is being recognized as a potential threat that is not truly a potential threat, and an immune response is elicited. Again, what categorizes each of the 4 types is what specific response is taking place in those components.